Welcome to the Deep Dive. Today we're tackling a really interesting concept: hyperbaric oxygen, HBO, pretreatment and preconditioning. So imagine breathing pure oxygen at, well, higher pressures than normal before something challenging happens. Could that actually give our bodies a bit of an edge?
That's exactly it. And we've got some fascinating material that looks into just that. We're focusing today mainly on two areas where this pretreatment seems to show, real promise
Mature.
in the world of diving and also in surgical procedures,
Yeah.
specifically thinking about cardiovascular stuff.
Okay, so our mission today is to figure out how this simple act, really breathing pressurized oxygen, can trigger some pretty remarkable protective mechanisms inside us. All right, let's start with the diving science. Plunge right in.
Sounds good. So one of the big worries in diving, as you probably know, is decompression sickness, DCS. It happens when nitrogen bubbles form in your tissues when divers come up and the pressure drops.
Right, those nitrogen bubbles,
Mhm.
always the issue. So how does this HBO pretreatment help divers deal with that?
Well, the research points to a couple of main things. First, tissue denitrogenation.
Denitrogenation?
Yeah, basically washing out some of that nitrogen before the dive. So there's just less nitrogen around to form bubbles when they ascend.
Okay, that makes sense. Less raw material, fewer bubbles.
Yeah.
Simple physics, almost. What else? How else does it help?
Well, besides just lowering the nitrogen load, it seems HBO pretreatment also directly affects bubble formation itself. It actually reduces the number of air bubbles that pop up during decompression. And interestingly, it seems to help platelets, those blood cells involved in clotting, recover faster after a dive. Platelet activity changes can actually be part of the DCS problem.
Huh. So it's not just gas physics, it's also affecting how our body responds at a cellular level. Interesting. Were there any specific findings in the diving research that, really stood out?
Oh, absolutely. One thing I found pretty striking was that doing the pre-oxygenation underwater, say at 6 or maybe 12 meters, seems to work better at cutting down bubble formation than just doing it on the surface.
Really? Underwater?
Yeah. It suggests the higher pressure itself during the pre-breathing is a key factor.
Okay, so breathing the oxygen at depth is more effective. That does raise questions about the pressure component itself, doesn't it?
It does. And further to that, the research shows breathing pressurized oxygen, that's HBO, is definitely more effective than just breathing oxygen at normal sea level pressure, normobaric oxygen, or NBO. It works better for reducing both the bubbles and that platelet activation we mentioned. So the pressure seems to add an extra protective layer.
And it sounds like these benefits go right down to the cellular level, even our immune system.
They do seem to. Oxygen preconditioning in water was shown to boost the antioxidant action of lymphocytes. Those are key white blood cells. And it helps lower levels of reactive oxygen species, ROS.
ROS. Those are the damaging molecules, right? Produced under stress.
Exactly. Think of them as like metabolic exhaust fumes that can cause damage. So the underwater oxygen seems to shield these immune cells, making them better at handling that stress.
So it's basically prepping the immune system for the dive ahead.
Precisely. It also seems to help these leukocytes keep a healthy calcium balance, which points towards better function under pressure. Researchers even saw more activity in the genes that make antioxidant enzymes, things like catalase, superoxide dismutase.
Wow. So it's like telling the cells to ramp up production of their own internal protectors.
Kind of like that, yeah. Boosting their defenses, like turning up the production lines for catalase and superoxide dismutase, these really powerful antioxidant defenders.
That's quite a preemptive boost. And you said no increase in intracellular calcium was seen.
Correct, which again suggests less oxidative stress is happening inside the cells.
Okay. Now, you mentioned washing out nitrogen at ground level earlier. How does that compare?
Well, there's an interesting point there. Breathing normal oxygen on the surface does help clear nitrogen, but that benefit kind of gets neutralized once you go down to depth. However, HBO pre-breathing, say at 1.6 atmospheres absolute, was still found to cut down on bubbles after a dive to 4 atmospheres.
Even after a deeper dive.
Yeah. And this seems to support the idea that HBO helps clear out tiny gas nuclei.
Gas nuclei, like microscopic starting points.
Exactly, like seeds for bubbles. So by getting rid of these tiny seeds beforehand, HBO makes it harder for the bigger, troublesome nitrogen bubbles to form and cause DCS.
Got it. And how long does this reduction in nuclei last?
The research suggests it's quite variable. The time for these nuclei to regenerate could be anywhere from a few hours up to, well, maybe 100 hours.
Wow, that's a huge range.
It is, and it has big implications for planning dive schedules, safety protocols, that kind of thing.
Definitely a significant window.
Yeah.
It really shows the potential power here. I also saw something about just breathing normal oxygen before a dive making a difference, like even for 30 minutes.
Yes, that's true too. Even just 30 minutes of breathing regular oxygen before an open water air dive has been shown to significantly reduce venous gas emboli. Those are the bubbles we can detect in the veins post-dive.
But the sources also warn that doing more dives increases bubble production and DCS risk anyway, right?
Oh, absolutely. Pretreatment helps, it lowers the risk, but it's certainly not a magic bullet. You still need caution.
Right. So the sources mentioned three main theories for how oxygen pretreatment reduces these bubbles.
Yeah, they broke it down into denitrogenation, which we've covered, getting rid of those gas nuclei, which we just discussed, and then hemodynamic effects.
Hemodynamics, so effects on blood flow.
Exactly, how it affects circulation.
Okay, let's unpack those last two a bit more. The gas nuclei removal is pretty clear now. What about this idea of tissue nitrogen supersaturation actually preventing bubbles? That sounds backward.
Yeah, it relates to the oxygen window principle. It's a bit complex, but basically our bodies constantly use oxygen. This creates a slight pressure gap where the total gas pressure in our tissues is naturally a little lower than the surrounding pressure.
Right.
When you breathe high oxygen concentrations, this gap, the oxygen window, gets wider. This makes it harder for inert gases like nitrogen to reach the high supersaturation levels needed to actually form bubbles.
Okay. So the higher oxygen level creates more room, essentially, making it harder for nitrogen to bubble out. A safety margin.
You could think of it like that, yeah.
And how does the oxygen actually get rid of the gas nuclei? You said it displaces the nitrogen.
That's the theory. The high partial pressure of oxygen during HBO lets it diffuse into these tiny microscopic gas pockets or nuclei. It pushes the nitrogen out, essentially, causing them to shrink or maybe even dissolve completely before they have a chance to grow.
Wow, oxygen physically pushing out nitrogen at that tiny scale. That's amazing.
It is pretty neat.
And I think I saw something about the lymphatic system, too, helping clear protein-coated bubbles.
Yes, there's some suggestion that oxygen might help the lymphatic system, part of our waste clearance network, remove these tiny protein-coated nuclei. It might not reduce the actual number or density of nuclei, but maybe it reduces their size, which could also lower DCS risk.
Interesting. Okay, and finally, those hemodynamic effects. How does breathing oxygen change blood flow?
Well, studies show that even an hour of oxygen pretreatment at sea level can lower your heart rate and cardiac output. And at the same time, it increases vascular resistance, kind of tightens up the blood vessels a bit. These effects can last for over an hour.
And how would that affect bubbles?
These changes in blood flow could influence where bubbles form, how big they get, and how they're transported around the body. The exact impact is still being studied, but it's another potential mechanism.
Okay, deep dive into Dyes Incomplete. Let's surface and switch gears. The other big area was surgical procedures. It sounds like HBO pretreatment might have some, well, equally surprising benefits in the operating room.
That's right. The early results from using HBO pretreatment in clinical settings, especially before big surgeries like cardiopulmonary bypass or major pancreatic surgery, have been quite encouraging, even if initially the exact why wasn't totally clear.
And a major risk in those surgeries, and also things like, say, a blood clot or using a tourniquet, is this thing called ischemia reperfusion injury, IR injury. What's going on there?
Okay, so IR injury happens in two phases. First, ischemia. Blood supply to a tissue or organ gets cut off. Then reperfusion. The blood flow is suddenly restored. Now, restoring blood flow is obviously good, essential even, but that sudden return can paradoxically cause damage.
How so?
You get a rapid burst of damaging molecules, oxidants. You get a release of inflammatory signals, cytokines, and those reactive oxygen species, ROS, we mentioned earlier, like hydroxyl radicals, hydrogen peroxide, nasty stuff. It can also trigger the complement system, part of the immune response, which can sometimes go overboard and damage healthy tissue.
So the fix itself causes a new problem. Ouch. And our sources said different organs handle this differently.
Yes, some are more vulnerable than others. Lungs, liver, kidneys, brain, heart muscle, testes, skeletal muscle.
Yeah.
They're all susceptible, but the degree of injury can vary.
Let's focus on cardiac surgery for a bit, since that came up a lot. What are the specific dangers of IR injury during something like bypass surgery, where the heart stopped and a machine takes over?
In cardiac surgery, IR injury can mean the heart doesn't pump as well right after the operation. That's reduced cardiac output. Longer term, it could contribute to heart failure. You can also get myocardial stunning, where the heart muscle is weakened temporarily, or even actual cell death, necrosis, or apoptosis.
And the brain is a big worry, too, during these heart operations, right?
Absolutely. Brain injury after cardiac surgery is unfortunately a known risk. It can range from subtle things like cognitive issues or delirium after surgery to much more serious events like a stroke.
What causes that?
It's often multifactorial. Tiny clots called microemboli, maybe reduced blood flow to the brain during the procedure itself, inflammation, issues with temperature control during surgery, even genetic predisposition can play a role.
Okay, that's a lot of potential risks. So how does this idea of preconditioning fit in? It sounds like trying to train the body beforehand.
That's a good way to put it. Preconditioning is all about triggering the body's own internal defenses, making cells more resilient to that IR injury when it happens.
And how might HBO preconditioning do that? Breathing oxygen before surgery. How does that help protect against this reperfusion damage?
Well, the thinking is that HBO preconditioning might stimulate the production of protective antioxidant enzymes, similar to what we saw in diving, things like catalase, superoxide dismutase, maybe hemioxygenase 1, especially in the central nervous system. It's like the HBO sends a signal, hey, get your defenses ready, something might be coming.
So priming the cellular defense systems, what results have actually been seen in hospitals using this?
There are some promising clinical studies. For example, one found that giving patients 3 HBO sessions at 2.4 atmospheres before their on-pump coronary artery bypass surgery actually led to less neuropsychometric dysfunction afterwards.
Meaning better brain function after surgery.
Yes, fewer cognitive problems, and it also seemed to help control the body's inflammatory response.
That's potentially huge, reducing brain complications after heart surgery. Any other benefits observed?
Another study looked at just a single 90-minute HBO session, this time at 2.5 atmospheres, before on-pump CIBG. They found it improved how well the left ventricle, the heart's main pump, worked after surgery. Patients also had less blood loss, shorter ICU stays, and fewer complications overall.
Wow, that's quite a list from just one session beforehand. And it might even help prevent arteries from re-narrowing after some procedures.
There's some suggestion, yes. Evidence that HBO might help prevent restenosis after PTCI, that's angioplasty, particularly in patients who've just had a heart attack.
And can we actually measure this protection, like in blood tests?
To some extent, yes. Preoxygenation has been shown to lower the levels of certain biomarkers in the blood around the time of surgery. Things like S100B protein and neuron-specific NLAs, or NSC, those indicate potential brain stress or injury. And troponin I, which signals heart muscle stress, lower levels are generally better.
And doing repeated sessions seems even better, at least for some surgeries.
It seems so. One detailed study found that repeated HBO preconditioning significantly reduced the release of those brain markers, S100B and NSE, in patients having on-pump bypass surgery, which suggests, better outcomes for both heart and brain function.
But not in off-pump surgery.
Interestingly, no, they didn't see the same benefit in off-pump CIBG patients in that particular study. This might mean the HBO is specifically helping counteract stresses related to using the heart-lung bypass machine itself.
Ah, that's a subtle but important distinction. The context matters.
Definitely. It highlights how specific the effects might be.
And didn't the sources mention a specific mechanism in the liver as well?
They did. One animal study pointed to the activation of an enzyme called hemoxygenase 1, or HO1, in the liver as being really important for the protective effects of HBO preoxygenation against liver IR injury, which suggests maybe similar protective pathways involving HO1 could be at play in other organs too.
So it sounds like the potential is definitely there, but we still need to figure out the best way to use it.
Exactly. The researchers are clear that more work is needed. We need to nail down the optimal parameters, the best pressure, how many sessions, the timing relative to the surgery for different situations.
Okay, so let's try and wrap this up. To recap our deep dive, hyperbaric oxygen pre-treatment and preconditioning seem pretty promising in, well, 2 quite different fields. It helps divers reduce the risk of DCS, and it potentially protects patients, especially those having heart surgery, from ischemia reperfusion injury.
Yeah, the core idea is really fascinating, isn't it? Using oxygen under pressure proactively to switch on the body's own defenses, whether that's bracing for pressure changes in a dive or for the blood flow disruption during surgery.
It really is. And it makes you wonder, given these findings, what other situations might benefit from this kind of approach? Could we see this used elsewhere in medicine? Maybe even, high-performance sports? That's definitely something for you, the listener, to think about. Thanks for joining us on this deep dive.